Good evening. Good evening. One more time. Good evening. And welcome to Shoals Marine Laboratory's weekly marine science seminar. I'm Sarah Morris, the John M. Kingsbury Director of the Shoals Marine Lab, and I want to thank you for joining us this evening for our 11th Rock Talk of our 2024 season. Our seminars are affectionately known as Rock Talks because when the lab began in the 1960s, the lectures literally happened in the intertidal on the rocks. Although we've moved inside, which is way better during thunderstorms, we still use the term Rock Talks because it is a wonderful connection to our island community and to the history that we have here at the Shoals Marine Lab. For those of you that are joining us online who may be new to our lab community, the Shoals Marine Lab is the oldest and largest marine lab in the country that is focused on undergraduate training, both in terms of coursework and in research. The lab is operated jointly by the University of New Hampshire and Cornell University. In addition to our guests online that are joining us remotely, we have 3 courses on Appledore: Research in Biology, Marine Environmental Science, and Introduction to the Biology of Sharks, Skates, and Rays. We also have our Shoals Undergraduate Research Group, affectionately known as SURGES, who are in the process of developing their own talks for our symposium this Saturday. We're really excited to have everyone, both in person and online, joining us this evening. The format for tonight's talk is a 45-minute talk, followed by 15 minutes of questions and answers. For our friends online, please use the Q&A button at the bottom of your screen. And for our audience here in the Commons, we ask that you raise your hand. I'll bring the mic around so that everyone, both online and in the room, can hear you. I would now like to invite Dr. Rebecca Atkins to introduce this evening's speaker.
Thank you, Sarah. I am excited to welcome this evening's speaker, Dr. Mike Sigler, who is a scientist in residence here at the Shoals Marine Lab. Mike completed his bachelor's degree in animal science and his master's in veterinary medicine at Cornell University. He then completed a PhD in fisheries science at the University of Washington. Between then and now, Mike has accrued over 30 years of research experience in marine ecology and fisheries stock assessment with the U.S. National Oceanic and Atmospheric Administration's Alaska Fisheries Science Center in Juneau, Alaska. There, he led the Habitat and Ecological Processes Research Program, studied stellar sea lion prey and predation, assessed Alaska sablefish populations, and developed quota recommendations, some of which he will touch on tonight. He also communicated his science back to resource managers and served on several state and federal fish stock assessment review panels in New England, the Pacific Northwest, and Alaska. His collaborative work even extended to advising scientists in Portugal and New Zealand on survey methods and population models. Throughout his career, Mike has contributed substantially to our understanding of marine ecosystems by co-authoring more than 60 peer-reviewed publications, 34 large-scale research plans, and writing assessments and technical reports. He strongly believes in the practical value of ecosystem research and its application to the management of fisheries and the conservation of seabirds and marine mammals. Mike is also setting the bar for what it means to be retired, which he supposedly did back in 2017. Despite that, he still collaborates with colleagues at NOAA and Canada's Department of Fisheries and Oceans to deepen our understanding of marine ecosystems. It also probably goes without saying that he is incredibly generous with his time and is passionate about sharing his experience and the value of applied science with students through teaching and mentoring. We are proud that he is among the core faculty at Shoals, co-teaching the Marine Ecosystem Research and Management course, and I'm also incredibly lucky to work alongside him for 10 weeks during the summer as an on-island mentor to the Shoals undergraduate research. When he's not doing full science and mentoring at Shoals, Mike can be found lecturing at the Friday Paper Laboratories in Washington, extensively exploring the outdoors on his bike, and most importantly, helping flora in the garden. It is my pleasure to introduce my colleague, good friend, and Flora's husband, Dr. Mike Sigler, to present his talk, Marine Ecosystem Research and Management. Please join me in welcoming Mike.
Good job.
Rebecca stole my line about being better known as Flora's husband. So what I want to try to do is tell you about my professional path and how I evolved from somebody who was focused on single species to multiple species and then eventually studying whole ecosystems, and try to convey to you a few stories or lessons I learned in doing so. So Mike's path, what is marine ecosystem research? Part 2. And in part 3, I'll talk about an example of species-centered ecosystem research. So this is the young Mike with hair, front and center on the common stairs, 1978. I went to Alaska in 1982. And then, as Rebecca mentioned, it's their older son, Andrew, earned my PhD in 1983 in Washington. The first chapter is talking about the single species part, Alaska sablefish stock assessment from 1983 for about 20 years. And I was part of a group. Our task was to ensure the sustainability of a $100 million fishery. What's a stock assessment? Basically, how many fish are in the ocean and what is a sustainable harvest to derive from that population. So the first thing I want to say is Flora is displaying the best tasting fish in the ocean, sablefish. This is in a restaurant in Honolulu. And to tell you a little bit about the fishery, the Alaska sablefish fishery has long been a bottom longline fishery. In other words, there's buoys to the top, buoy lines to the bottom, anchors, and then several miles of line with hooks, baited hooks strung along. There's 2 points I want to emphasize. First, it's a single species. This is when I'm a single species scientist. And the second point I want to make is there's lots of research necessary to support a reliable stock assessment. And I'll give you 3 examples. And typically what I'm going to do here, I'll show you a paper title, the study purpose on your left-hand side, and then some nice pictures. That's a sablefish hooked on a bottom long line here observed from a man submersible. So the first thing we want to do is understand the accuracy of our tool for measuring fish abundance. So I spent about 125 days at sea conducting field work using different kinds of technology, man submersible, a device called a hook timer, to evaluate the accuracy of our tool for measuring fish abundance. You need that thermometer on the population to get a good measure of the population size and its changes. You also want to know the vertical availability, or in general, the availability of the population you're measuring. This is an archival tag in the hand, and then a pattern of diel vertical movement over 40 or 50 days for the sablefish, going up and down between 300 and 600 meters. That tells us something about how available our population is to our samples. It's also a little bit of biology. Once we have data, it's not just the survey, but other information like the age composition of the stock of the population. We want to evaluate our tool for estimating how many sablefish in the ocean. It's typically done with something called an age-structured population model. This shows a time series of estimates, and likewise, I did a paper to evaluate how well that population model does with sablefish. A couple things I want to point out about the life of being a stock assessment scientist. Populations go up, they go down. And you can see a 60-year time series. This is from the most recent assessment. Mike worked on it during the red rectangle. And you can see an earlier period of overfishing, subsequent to stock rebounded, a subsequent decline in the population, in this case, due to low juvenile survival. So in some sense, you have to land the plane, reduce quotas to match where the current productivity of the stock is. is located. And you want to jigger those quotas down until you settle it down, which can be fun. It's both fun and tricky. I want to leave 3 points of view about stock assessments. One is like when you do all your projects in the island, you need an accurate sampling tool. You want to be able to measure your organism's availability to that sampling tool. And then whatever kind of data analysis you do, you want it to be a robust estimation. So after a time, after about 15 years, I transitioned from single species to ecosystem research. And I had 2 reasons. One was I wanted to better understand why populations wax and wane, why they go up and down. And I also wanted to work with the new species. I basically wanted to stir the pot in my brain. Not quite the first one I did, but I'll first talk about this, the Bering Sea project, which occurred from 2007 to 2015. It was a large project with dozens of scientists working on the project. The goal of it was to understand climate effects on the Bering Sea, from zooplankton to phytoplankton up to fish, birds, mammals, and people. My focus in it was to understand 2 things. One, the picture shows the coast of Alaska. The Aleutians are trailing off towards the bottom. You can see sea ice extending from the coast, that west coast of Alaska. And my focus was on seasonality of phytoplankton and the subsequent food web effects, as well as spatial relationships among predators and prey. And I'll lead to this point, but the dominant factor dictating what happens to the Bering Sea is sea ice. Sea ice rules the Bering Sea, seasonal sea ice. So likewise, a paper, that looked at how the spring bloom and the fall bloom timing is affected by climate change. Climate change occurs through loss of sea ice in the Bering Sea, both when the sea ice is there and when it's lost and where it's located. That affects phytoplankton bloom timing in the spring. I also looked at how loss of sea ice affects on zooplankton and other fish, food web effects as you go up the food web. This is a synthesis paper from the project. And then another paper was to look at spatial relationships among predator and prey. This is how I got to work with my first 2 seabirds. The take-home message here from our project was, as I said, sea ice rules. It dictates that loss and the change in its location drive changes in abundance of plankton, fish, birds, and mammals, and had subsequent effects on people who live on the coast, people who remove fish for commercial fish. My retirement is, Rebecca said, I'm teaching here, Friday Harbor's lab for a bit in the fall. I also coach little kids, Nordic skiing in the winter, all ages, the full age spectrum. So part 2, what is marine ecosystem research? You can answer questions such as, how does climate change affect the Gulf of Maine ecosystem? You can try to tackle questions such as, how do seabirds, such as the common tern, locate prey in the Gulf of Maine? And this is the second lesson. The first one was about single species stock assessment, is to talk about 2 basic approaches to marine ecosystem research. One is ecological process research. So you're trying to understand how some process in the ocean occurs and then how that affects the whole of the food web. You understand those effects. For example, looking at how lots of sea ice affects ice algae on the underside of the sea ice. Ephods love to eat ice algae. And then other fish in turn, like walleye pollock, to consume the krill, and they have effects on their predators. So So you're studying the whole of the ecosystem to look at this one process that's lost in the ice effect. Second approach is what I call species-centered ecosystem research. So for example, a Gulf of Maine example is that currently herring abundance is low in the Gulf of Maine. It's a sustainable fishery and wildlife conservation issue. If we want to study it, you could study it as a single species and at least just keep track of it. But the argument here is to do ecosystem research around it to understand why it's changing. So we study the ecosystem to understand why the prey effects occur. Predators, what's their effect on that and the herring? And then predators, including birds, seals, and fishermen. In this way, we can provide ecosystem understanding that addresses the conservation issues. And because I was agency, we also in turn provide advice to managers based on that science, ecosystem science. So what are the tools of the trade for ecosystem research? Any guesses? Hammers, screwdrivers. In the ocean. Nets, Micaiah. Or maybe it's not Micaiah. Sorry. Pardon? Maybe I'm getting a little deaf too. Yeah. Well, yeah. Okay. These are specific. But sorry. Yeah. Anyways, I like to think of there being three things that science depends upon, and this kind of framework helps to bring along a larger group of people to understand how to do work. So first you want to monitor what's going on. I'm actually not being specific yet. I will be specific. You want to understand why is something changing. And then if you know enough, you can predict or forecast. And this approach helps organize people from different disciplines so you can gather a whole research group together and get them pulling the cart in the same direction. You want to tackle ecosystem research, you have to think about things like that. So now the specifics, my apologies for first we want to do things like monitor ocean conditions, monitor abundance of things like zooplankton, monitor reproductive output. It's something that's very sensitive on an annual basis and is a good way to reflect what the climate is doing to the ecosystem. And I'm going to make one jump. One takeaway is that if you have enough monitoring, you can start to construct understanding. My colleague Phyllis Dabineau has a rule, she's an oceanographer in Alaska, that once you have 15 years of monitoring data, you can write a paper and understand climate effects. So if you look at the years on this, you see that rule was upheld in this case. And there's a little bit of thinking about that. You want to have climate acts on an annual basis, it's a cold year, it's a warm year, or whatnot. You essentially get one data point per year understanding climate effects in this way. To understand, you can do field studies. They're directed process studies. I'll spend some time towards the end of my talk describing one in more depth. We can have lab studies to the same end. For example, looking at temperature effects on a range of species that co-occur and see who will be the winners and losers in a certain region. We can use computer models to simulate an ocean and try to understand effects as well. They can be simple multi-species models, or they can be full ecosystem models that are spatially explicit. And then the last point, you have monitoring, then understanding. Once you have understanding, understand how the system works, you can start to try to make predictions. And when you do a project for class, you're essentially testing your prediction. You observe something, you do a little experiment, and then you're trying to make some kind of prediction about what's going to happen, and you test with that idea that you have. Likewise, once we have enough understanding, we can construct computer models to forecast what's going to happen. In this case, this is an example of some Stanland predictions for the next 30 years. And then we want to apply all this, again, as an agency scientist, conservation, wildlife conservation, fisheries conservation, and sustainable fisheries management. And so my role as an applied scientist, I viewed the managers and fisheries, the conservation managers, fisheries managers, in some sense, as a working scientist, they were my customers. So I'm just trying to provide them information. So when they make decisions about the ocean, they have information, credible information about what's doing that. And that's how you enable ecosystem-based management. You need that information to support treating the ocean as a whole, the Gulf of Maine as a whole piece, to make those recommendations, those changes. Obviously, it's a much messy process. There's lots of different interests and so forth, but at least we can provide information when there's decisions made to inform those decisions. And I'm going to make one other point. It's hugely important to communicate. It's hugely important to collaborate. You need a village to do this kind of work. And then I'm going to add and elaborate on it a little bit later. Take a no surprises approach when you're doing, especially work that might be about a controversial issue. So part 3. Oh, good. It's an example of species-centered ecosystem research, Sigler sea lion. And what I mean is research about the whole of the ecosystem with stellar sea lions at the center. The research rationale was to understand a decline of western stellar sea lion in the western part of Alaska, as opposed to eastern stock where eastern part where Florini live. And for many years, there's a dramatic decline in the population in the western part of Alaska. And it was declared endangered in 1997. The challenge was trying to manage its prey because the largest fishery by volume is an important sea lion prey. So you have competition in the fishermen and this endangered species for the polypus prey. And further time and area closures were proposed for the 2000 fishery to mitigate decline. And this provoked a political firestorm. And it prompted also substantial research funding. This is how I got my start in working on multiple species, because I bugged my boss so I could apply for funding to do the 2 studies I'll talk to you about. So I wanted to change gears, and I said, I want to work on the problem. It's of interest to me. I also want to diversify the work that I do. And this research wasn't just the 2 projects I'll describe, but a whole, I think there was like 30 projects that were developed to address this problem. And they tackled how environmental change was affecting potentially stellar sea lions, fisheries competition, like I just said, the pollock fishermen they prey on they're catching the same thing that the sea lions predate. Predators like killer whales, and other things like contaminants. World War II brought a lot of contaminants to Aleutian Island, where there are many sea lions, rookeries, and mollusks. And lastly, I worked on 2 projects, in part because there's a shark class here today. I'll show you a slide describing what we did about whether or not Pacific sleeper sharks related to the Greenland sharks found out here, predate stellar sea lion pups. There was some idea they might hang out near the rookeries, but the pups first go in the water and be predated by the sleeper sharks. And so we looked at the diet of the sharks around some rookeries. We also tagged them with pop-up archival tags, and they demonstrated this really cool oscillating behavior in the water column. Over 2 days, they went up, this particular individual went up and down the water column probably 30 times. And to me, it's amazing how rhythmic it is. Like it knew where it was in the water column. The other project that I'm going to talk more about is the Southeast Alaska Stellar Sea Lion Prey project. So this is on the other end. It's my favorite project in my career. Very fun. It was fun because I got to go out in the field year round, go out to sea year round, and see how things change from summer to fall to winter to the next spring. So I'm a skinny guy. I'm motivated by food. And I know where dinner is. The comments, right? Who thinks the same is true for a wild animal, an animal in the ocean? They know where dinner is. Raise your hand if you think they know where dinner is. Yeah, it's smattering in hands. Why do you think they know where dinner is? They're smart. And they can have memories, right? Okay. But what about those sneaky prey? What are they going to do? Not go there again. They might be smart, or at least, fish, marine mammals, I don't know. But it's a problem for wild animals to find prey. And what do they do to do that? And they have to balance the cost of foraging against the benefit of finding. So I thought about the importance of predictable prey. It's been demonstrated for many terrestrial species. Brown bear in a salmon stream in Alaska. Salmon typically come back in a relatively window so that bears, bald eagles, and everybody and their brother can come there, predate the salmon coming back. Predictability. But it hasn't been, and not that much applied to air-breathing vertebrates that forage at sea. And think about foraging. So if you had to swim around to find stuff, it's not just walking around a two-dimensional surface on land. You also are faced with the third dimension search. It's a value thing. Your sighting distances, you look underwater, if you can see 100 feet in the tropics, everybody goes, wow, that's amazing. Well, just think of we can see to the main spot. Sea lions can't do that, or other animals underwater can't do that. And the last thing is, fish have lots of very good reception to odors. Sea lions don't. So they don't have any smell sense well underwater. So think about the environment they're in and what are they going to do to try to find prey? Prey that runs away and doesn't stay in the dining day to day. Further, if you try to forage around, you might, and do it efficiently, you might get in the sights of the killer whale. So those are some of the things that an animal, a marine mammal, a sea lion in general will face. One message I want to pass is to think about, know your system. When you study something, think about like you're that organism. Okay, so for example, a stellar sea lion is a central place forager. They're tied to a spot, either when they're rearing pups or even when they're just falling out, because they typically fall out in certain spots, I think, to help them evade predators. So they're tied to a central place. So when they go out, they're going to come back to the same spot. Second thing, as we mentioned, their prey is migratory. So they have to, they're a central place, they're moving, know your system, think about that in context of the study of that animal. So some specifics, we did this Southeast Alaska Stellar Sea Lion Prey study from 2001 to 2005. Southeast Alaska in the red square there, Flora and I used to live right about there. It takes a village There were several collaborators. There was a mixture of marine mammal scientists and fisheries biologists and ecologists that we worked together to do it. Collaboration is great. You will get more power if you collaborate by working with other people. You write a better study, you'll write a better paper. So how do you do species-centered research? Well, to me, the approach is first, measure the available prey. How many fish are in the water? How do we find out what their diet is? Scats. It's actually kind of fun to go collect scats. I hate to admit it, but we measure the diet, what do they in fact select. So from the sweet animals that are in the environment, what do they in fact select? The last thing to do is track the location in some way. Tags would be one way, there's others. But those three pieces are important to do this. So to measure the prey available, we use a mixture of survey styles of measuring tools, mid-air trial and acoustics. And with those, we had concurrent visual scans to associate stellar sea lines. Where do you see the stellar sea line? Was it associated with the concentration of the prey or not? Because fish occupy many different kinds of habitats, we had to use different kinds of sampling tools. Nearshore beach seine, underwater camera. And then for demersal or on the seafloor, bottom long, the thing that I showed. So we're sampling all the habitats. We also measured nutritional energy of the prey. The reason was that we wanted to change biomass into a common currency. At some point, fish eat calories, they don't eat food. And so we changed everything and by getting measurements of nutritional energy, we translated all our biomass to calories. We picked up scats, used aerial surveys. So for 36 months, Jamie Wobble flew around in a fixed wing plane and took pictures of fallout. Sort of like going by Duck Island year round times 30 once a month. And it was actually super useful to evaluate, to extrapolate our findings from our narrower fishing area, study area. And then we had some satellite tagging where this is an older technology. Sea lions don't go on land. But this still gave us information on seasonal movements of sea lions if they follow seasonally available prey. The available prey are shown here. So we measured all these different prey types. Some fat fish, some lean fish, some nearshore fish, some really deep. And then the ones that aren't crossed out are the ones that we learned about. Yeah. And they tended to pick pelagic fish, and they often selected fattier fish, but not necessarily. Here's one piece of it. So we did monthly and quarterly and seasonal surveys. From our monthly surveys, singing on one area that had overwintering herring. So this big red blob, school of herring, millions and millions of herring, hunkered down for the winter. Actually just outside of Juneau. Nearby, there was this fallout that was occupied in the winter. 0 animals in the summer, 500 in the winter. So these sea lions would take advantage of that herring school. And from our study, we can compare the black line are the sea lion counts. Whoops, back one. The black line going up and down, and in tandem, the herring went up and down. So sea lion attendance at this hollow increased in concert with the seasonal increase in herring during the winter when they were on the seafloor, like shown in the upper corner. Do it once, do it again, and in the third winter, we found the same thing. So it's clear that the sea lions are moving to a seasonal hotspot of herring overwintering here. So we found one secret spot. We found one common that they use and that they remember and are smart about. This was the advantage of what Jamie did in the plane. We could relate the count in the aerial counts from the aerial surveys and see if you had similar up counts in the winter, down summer, back up the following winter for other haul outs, places where sea lions would haul out. And there were about a dozen where this pattern occurred and where we could, from anecdotal information, find out, yes, there are seasonal aggregations of herring at those same spots. So in this way, we were able to extrapolate our information and strengthen our. In honesty, it was a bit of serendipity that it came out that way. So why are herring more available in the winter? So typically, herring don't feed in the winter because there's few prey and they move little. So they're basically hunkered down on the bottom and they stay there in big concentration. They do get predated quite heavily by the sea lions, but that's the choice they're making in their own foraging, survival strategy. In the summer, it doesn't happen because then there's lots of prey for them. Zooplankton are out and about in the photic zone, and they're following in smaller schools these ephemeral catches of copepods. And so that seasonal aggregation of herring is no longer available to the stellar sea lion. They move on. So our main finding one from the study is that sea lions move among seasonal hotspots. I talked about herring in the upper right-hand corner, but there's similar stories that we discovered for eulachon or smelt. Availability in the spring, salmon in the summer and fall, and they're moving between these spots. And by the way, there was always this background species of pollock that was spread out everywhere, but not seasonally aggregated. So it's like they had these common places. They knew the food was really good, but they could always go out and maybe find some berries out in the bushes here, equivalently to the pollock. Second finding, and I didn't talk about this at all, but if you, once you translate on the x-axis all that biomass to food energy and plot it versus the sea lion counts from those aerial surveys, we can look at if there's a numerical response to the sea lions to increase nutritional prey. And that's what we did. What this helps us do is get some sense of what the minimum prey energy is to support, say, 100 sea lions. And it's not just that they have meals on the table, it's orders of magnitude more than that, because they have to find that prey first. It has to be a sufficient density for them to support successfully. The overall finding of all this work, so I described 2 studies. As I said, there was about 30. The overall finding was that nutritional stress was being focused, something about the prey. So what did the government do? They have to respond to the endangered species listing. They can't do anything that will make it worse. So it was put in place. There'd already been some time and area closures of fisheries for protecting sufficient sea lion prey. And I would say, just as an aside, it actually took years and years to get to that point where they made the places big enough. And it was lots of, probably 30 of my colleagues spent a lot of their time for 5 years working on it to hash it out in the management arena in the North Pacific Fisheries Management Council process. And the outcome, though, is good. Both stocks are increasing now. So it worked. Science plus management, decision making, and then implement management measures. So the last thing I'm going to talk about as a subject was something I worked on starting in 2012. It's on Alaska coral and sponge, and I'm doing it mostly to highlight the no surprises approach that I mentioned before. Because if you work as agency scientists or in areas as a consultant or whatever it is, in areas where it's controversial, water use in the West, saving fish, I'm going to make the argument for a no surprises approach. So, in the picture is Chris Rupert, a colleague of mine, a great person to work with, starting up the camera that was used drop camera that was used with stereo, 2 cameras, and dropped to the seafloor up to 800 meters in depth. It's a great tool. You can turn it over really fast, get 8 to 10 samples per day. And that was used primarily by Chris and others to identify deep sea coral and sponge communities and their distribution. So where are coral and sponges in Africa? I became involved, well, then the motivation of a lot of this work is to balance coral conservation with fish management. So if you, fishing gear can damage coral. So if we still want to remove fish from the ocean, we got to consider the damage of the coral and sponge. How do you do that? Close areas, change gear, or whatnot. So I started working on it when a group like Greenpeace and others called for protection of Bering Sea submarine canyon. And they mounted a public relations campaign, and several environmental groups allied themselves together to do so. In our job, a few of us were tasked with studying those Bering Sea canyons, seeing where the coral and sponge were in those areas. And in doing so, I'm not going to talk about the science, but I'm going to talk about the no surprises approach. So in developing the research, we were careful to take a no surprises approach. What do I mean by that? First, you engage stakeholders. Stakeholders, they're fishing groups, they're the environmental groups, they're any interested public citizens. You engage them during research development. Come to our meeting, we're going to talk about how to do it. Then once you do that, communicate regularly with them. Call them on the phone. You're in Seattle, I'm in Seattle, go meet with them or whatnot. Whatever it was it takes to let them know what's going on. And the last thing and the most important thing, don't let them find out about your findings at the management meeting. They will rake you over the coals. So you tell them beforehand, even if you know they don't like to hear the outcome, you talk to them, you have seminars, public meetings, whatever, to let them know before the decision-making meeting to answer their questions. It's super important. So I'm going to close with 3 takeaways. And first is to I talked about conducting ecological process research, for example, loss of sea ice, to understand in particular and often climate change effects. Lots of sea ice, look at how that affects the ecosystem. Duck species-centered ecosystem research, for example, stellar sea lions, tackle conservation issues, different flavor research. And then finally, a key part, especially for number 2, communicate, collaborate with people. It'll strengthen your project with a multidisciplinary team, and you work together in some ways in concert with the environmental group of fishermen, and then you carry out a no surprise. So that's what I wanted to say tonight. Thank you for your attention.
Thanks, Mike. And now he will take any questions from in the room or online.
Hello. So, I know in the projects that you were involved in, it seems like the people who manage like the fisheries and stuff like that were fairly cooperative. But Have you ever had instances where the stakeholders did not cooperate, even if you took the no surprises approach?
I would mention two examples. So when I was doing the stock assessment, it was during a period of decline, and so I had to recommend declining quotas, and fishermen were not happy, and they'd come up to me and get in my face. But what I would always do, I'd say, well, what were your two things happened. One is I spent a lot of time at sea, over 800 days at sea, so they could have some respect for me in that sense. The second thing is that I would just do them and say, what have your catches been last year, this year? And basically get them to tell me about what their experience is. And eventually I also incorporated their logbook information into the quantitative stock. So that was a way to diffuse that. The other case would be I reviewed the lobster stock assessments on the East Coast, and then New England is notorious for having difficult industry people. And I guess I didn't so much have to deal directly with it. And I was aware of it. There were some people at lobster fishermen at the meeting. And we just tried to do a good scientific review of the assessments, and that's how it went. Yeah.
How did you like living in Maine, just like as an experience, or in Alaska?
Great. Yeah, it was really fun. Beautiful place. The ocean's right there. The mountains are right there. I could go skiing at lunchtime. Kind of a nice bonus. It was a good place for us to raise our kids. It's dark. You got to get out. But to me, Southeast Alaska, the most beautiful place I've been. You know, and granted, it rains. Kind of got to say, oh, isn't that pretty the way the fog goes up the mountain? But yeah, it's a great place, I think.
Second only to Appledore.
Oh yeah, yeah.
I just want to know how long you've lived on the island, on this island.
On this island? Well, I've been here probably a total of a year, I suppose, something like that, going on a year. I mean, staying on this island, right? So I was a student for a month, kitchen for 3 months, and then a TA a month, so 6 months, and then 3 summers, 2.5 months each. So it's adding up. I know where the gulls are, anyways.
I'm from Ithaca, New York, and I was wondering, did you know when you were there that you wanted to go into like marine sciences, or did you kind of figure that out while you were there? It's not very, it's not a beach town by any means.
Well, I grew up around the St. Lawrence River, and we always camped as kids. I was a Boy Scout, so we're always outside. And when I went to Cornell, I wanted to be a veterinarian. Started to think maybe not. I came here, I was like, I just changed my mind, went in a different direction. Inspired. Laura's husband, nice to meet you.
This is trying to get me to go as far across
Yeah.
the room as possible.
Get up to a carrier pigeon.
Hi, Mike.
Hey, carrier.
Can I ask you two things about this interface of using your data to inform management? One was something that you just said in response to the other questions. You said that you incorporated some of the data, logbook data from some of the fishers that you talked with into your quantitative models. Can you talk a little bit more about that? And that, I think that just seems really like an important type of partnership. Could you elaborate?
Sure. For example, I went out with some of the fisheries observers on one of the boats that I knew, with the captain I knew, so I could see how data is collected by the fisheries observers. That was one step. And then that's when I started incorporating fishery observer information, the quantitative document. There was also an existing logbook program, but it was not incorporated into the stock assessment. So I got all that data scanned and put into the computer. And then I could take that data, analyze it, do the same thing. The last part was that boats under 60 feet are not required to have observers or logbooks, so they're unobserved either direction. So I worked with the Alaska Longline Fishermen Association, Boating Vessel Owners Association, Petersburg Vessel Owners Association. Did the same thing and set up in collaboration with them the voluntary logbook program. And then likewise got this information in the catch rate, so on and so forth, analyze the information, add it to the document. And it's kind of a two-way street. One is you build credibility by incorporating their information. And you also, I mean, fishermen don't fish the same way if you did the survey, okay? Because they go to the hot spots, which might not be sensitive to changes in population. Fish might always stay in those secret spots because that conditions are tested. But you can still analyze for that, test for that, basically inside the model and extraneous to the model for correlations to and see where they diverge under what conditions. So it's cooperation. It gives the fishermen a stake in the game. That helps a lot. And it also strengthens some of the scientific investigation of the stock assessment as a scientific process.
Okay, Mike, you've got an online question.
Sure.
Sue Kennedy has given you one that you're going to have to be very careful in how you answer. What has been your most interesting project during your summers on Appledore?
Houston toes or Transect 23. I can't remember which. I mean, I can't decide which one.
What kinds of questions or what kinds of classwork and other experiences would you recommend to students who are now interested in quantitative ecology and other fisheries realms?
I think that the two things were helped me. One is having lots of hands-on experience. So I knew when I collected data, I knew the quality of it, and I could collect it and think about it in a scientifically defensible way. And it's not just like the narrowness of collecting it. It's also thinking about things like replication and what things affect your results. So for example, often how much you sample location-wise will dictate how good of an answer you get. Because that location is often the biggest variant. Fish abundances, or sea lion abundances, or zooplankton abundances. So that one side, getting hands-on experience through classes, anyone that will give you those kinds of experiences in class is helpful. It's like Shoals was helpful for that. Whatever you're doing in high school or any of these classes, it's all hands-on. They're trying to replicate some kind of project-based experience. Then for the education side, any kind of applied statistics is helpful. Trying to and also population modeling is helpful. And I would say, like among those, I would strain more towards the applied statistics side, population ecology, as opposed to when you take intro stats, they talk about feed tests and they tell you how it's derived, blah blah blah. Okay, but trying to understand those tests and how they work and how you can apply them. Be applied is better. I know the last point I would make. These days, there's so much information out there that's spatially referenced, but taking anything that can help you understand how to map stuff and understand where things are and why they are and doing that in a spatial sense. That's super helpful.
Do you have a project that you would like to go back on because you have like more questions? Or is there another area of research that you think that is really interesting that you would like to do more on?
Interesting. I can tell you one little regret. So when I worked on stellar sea lions, I mean, I called it an ecosystem study, and it mostly was, and partly that was my first multi-species project. I really wish I had done more I had done some oceanographic scanning to try to understand how the ocean was affecting, for example, there's cuts, like when the bloom occurs, when fall overturn occurs, and it ends the growing season. And that is actually probably a trigger for when the herring go to the bottom. And if I had done any oceanography, I might have understood it. So that is a regret that I wish I had done it.
Oh, okay, last one, I guess. Thank you, Mike. That was a
Sure.
great talk. I wanted to ask about indigenous communities in your like reaching out to stakeholders and or applying management. I know sometimes certain rules for fisheries may not apply to indigenous communities. I'm curious if you had interactions with that at all.
Yeah, during the Bering Sea project. So I would say one thing about Alaska is that people live and they hunt seals, they hunt whales, because if they can't, they won't be able to live in those communities. So they depend upon subsistence harvest to stay there. And in the Bering Sea project, as part of the project, and I co-authored a paper with some coastal community people from St. Lawrence Island. I'd have to check again. George Nungwook and others, and we worked on like subsistence use in these communities, and we worked on, for example, looking at bringing physical oceanographers together with ice biologists, George and other people from his community to look at how sea ice conditions affected walrus. So they were part of all our science meetings, the community members, and a direct part of it. Yeah.
Do we have any other questions? If not, I want to thank Mike tremendously for stepping in today. It's nice to know that Flora's husband knows a few things. And it's really nice to see how different phases of a research career can really provide a very rich and detailed understanding of ecosystems and kind of thinking about the big pictures. Trying to do just one small piece wouldn't give you the same level of understanding as looking at that ecosystem level. So again, thanks, Mike, and thank you for everybody who is here this evening. With that, we have one final Rock Talk next week. Join us for Jen Mixis Olds, who'll be talking about bioacoustic monitoring here in the Gulf of Maine.